difference between infecting one species compared to another species. Okay, so when it comes to naked viruses, why do they call them naked viruses?
Because they don't have an envelope.
Now we know. Okay, it's it's a nice word. Anyway, the naked viruses, when they enter the cell, how do they enter it? Yes. Or lies the cell membrane, pore entry or lysing the cell membrane. Okay, so what's the difference between the envelope and the virus and the cell membrane? Because we know viruses acquire envelopes from the cell membrane, but how is it different, the envelope from the cell membrane? Do we know? Anyone? Yes.
Does it have to do with like the plasma membrane proteins in that they can, the envelope, they can rearrange them?
They can substitute them. Yeah, they can substitute the cell membrane protein with their own proteins. The lipids are almost the same, the cell membrane and the virus. Okay. Now, did you receive my email in relation to the question? Annie, where's it?
She's right there.
Okay, that's she asked. Do you have any comments related to it? Okay. The other things, what do we know about retroviruses? I mean, why do they, how do they convert the RNA to DNA? Reverse transcriptase enzyme. Okay. And finally, what is virus tropism? Can somebody tell me what virus tropism is? Yes?
It's the capability of the virus to attach to a certain organ or cell type.
Okay, that's targeting certain organs, tissues, or cells. For example, the FIV targets CD4 positive T cells because it has the receptor for them. Sorry, the protein that binds to the CD4 positive T cells. Okay, so today we're going to continue this, finish it hopefully if we have time, and then we'll start the next lecture with canine diseases. Okay, how do we assess virulence? How do we know that a particular virus is virulent? You should know that these definitions actually are for lab animals, because there's a lot of variation between animals in terms of the response to an infection, the variability between species in terms of how to, again, respond to the infection. And therefore, it's very difficult to define virulence in outbred populations. This is why they use, to define virulence, inbred mice and inbred rats. The reason for this is because they uniformly have similar response to the virus or viral infection. So we can assess virulence by the lethal dose 50. What this means is the dose of the virus or the amount of virus that can kill 50% of the animals. Now, we're moving away, or we've moved away from defining the virulence in this way for ethical reasons. And so this can really only, this is done only for lethal diseases or lethal viruses that cause lethal diseases. The second one is infectious dose 50, and that's the dose of the virus that can infect 50% of the animal. So when it comes to applying this, if you want, to larger animals, cows, equine, horses, sheep, etc., it's very difficult to assess virulence of a virus. We can only assess it by what it does to the population, of animals that the virus is infecting. The ratio is very good indicator of virulence. Again, this is in laboratory animals, which is the ID50 to LD50. So in laboratory, when assessing virus virulence, they use these three definitions. Now, what is the basis of the variability among individual, if you want, animals, and between species? The variability is due how the animal respond, and that's due to what's called the major histocompatibility antigens. Do you know what MHC is, major histocompatibility antigens, in immunology? Yeah, you got this, okay. So basically those are glycoprotein that are displayed on the surface of cells, and they determine how the response, the immune response, the nature of the immune response is going to be. And we know that they play a major factor because if we use inbred mice of a certain haplotype, they respond differently from another haplotype. Take the example of mice of the H2B strain, the H2B strain, their resistance to influenza, H5N1. Whereas those that have the H2D mice, H2D haplotype, they die from the infection. So the MHC, which is represented here by the H2 in mice and HLA in human, and in other species like the bovine is POLA or OLA, those are really crucial factors that can determine how we react to an infection, whether it's a human, animals. It's important factor that determine how we respond to an infection, whether it's viral, bacteria, or other. Now, we talked about virus tropism, and virus tropism, we said that it's the ability of a particular virus to target tissue, cell, or organ. Now, take the rabies virus, for example. The rabies virus can bind to two types of receptors, and that allows it to be to have more wider tropism. So, for example, it can affect myocytes, it can affect salivary glands, it can affect the neurons. So, by this, we say that this virus has wider tropism. Now, there are other physiological factors that are important. Obviously, the two extreme, the age, which is the young and the old. The young has an immune system that is still developing, acquiring, if you want, the experience throughout its life. The old, we know that age or old age is associated with a decline in the immune response. And that decline makes them more susceptible to diseases. Stress is a very important factor. Stress, for example, during pregnancy, can result in herpes infection. Stress also can convert a subclinical infection to clinical infection, clinical symptoms, subclinical symptoms to clinical symptoms. And we know that stress is due to a number of reasons, including crowding of animals. The cell cycle stage also is an important physiological factor for some viruses. For example, in the case of parvoviruses, they target rapidly dividing cells such as the fetus, such as the bone marrow cells, such as the crypt cells in the intestine, hence the diarrhea, etc. So basically, they target those cells, and one of the reasons is because they have DNA polymerase, a lot of it. And so they exploit the fact the DNA polymerase is abundant to replicate their own DNA. Now, fever also is a physiological factor that can affect host resistance. And we know this, that the use of drugs, salicylates, or the use of corticosteroids, those are anti-inflammatory, but at the same time they make the situation worse. Their fever is, we know that most of the viral infection, if not all, in animals are associated with fever. So an exception, you could say the fish, there is no fever in fish. When it comes to fever, it's a natural response that can actually limit viral infection. So when it comes to using anti-inflammatory substances or reducing the fever, some subclinical, again, infection become more obvious, more clinical. And herpes, for example, virus infection flourish when corticosteroids and an agent that reduce fever are present or are used. We know what are the routes of virus entry. There are several routes. Now, these routes obviously they're similar to bacterial infection, though they can be a little different. When it comes to viruses, one mode of introducing viruses are mosquitoes and ticks. And normally the epidermis, we know that is thick enough to prevent any bacterial and any viral infection. However, abrasions can occur, whether in the field or naturally, contact with animal bites, etc. So once the epidermis is compromised, then it's likely that viral infection can be introduced into the host. But mosquitoes are one vehicle for viruses to enter the bloodstream. And we know many examples of mosquitoes that carry viruses. For example, West Nile virus. Other examples are the encephalitis diseases, toga viruses that also can be carried by mosquitoes, and a number of other also viruses. Viruses can enter the conjunctiva, and we know enteroviruses, adenoviruses can enter the conjunctiva and spread from the conjunctiva to other places systemically. And the usual one is the mouth. Now, when viruses enter the mouth, they're really faced with a number of barriers. The most obvious barrier is going to be the acidity of the stomach. Many viruses get killed by the acidity of the stomach, whereas other viruses can resist the acidity of the stomach, reach the intestine, and causes an infection in the intestine. Now, the respiratory routes is the most obvious, most important, if you want, route for viruses to enter the host, and others like urogenital tract. And others. So we start with the respiratory tract. That's the most common route. In the respiratory tract, the defense mechanism usually it starts with the epithelium itself, the epithelium with the cilia, the movement of cilia, make it difficult for viruses to attach to the cell. But if this doesn't help, there is another defense mechanism, the NALT. And the NALT is a nasal-associated lymphoid tissue. Nasal-associated lymphoid tissue. Basically, there are lymphocytes where they, in addition to lymphocytes, there are macrophages, there are dendritic cells. So in these, if you want, nodules or lymphoid tissue that is scattered all over the small intestine, there is a way of dealing, with the virus before it enters the subepithelial tissue. But sometimes this doesn't work, and viruses can compromise, if you want, the integrity of the epithelium, and hence they can enter the subepithelium into the blood circulation.
Dr. Mishra?
Yeah.
What does the B in the bulb mean? The B in the bulb?
Oh, the bronchi. Bronchi associated lymphoid tissue. Because what happened, obviously, if they cross the nose barrier and go down to the bronchi, to the lung, then there is a similar lymphoid structure, or there are similar lymphoid structure which is called the bronchi associated lymphoid tissue. If they go to the lung, all the way to alveoli, then they're faced with another barrier, which is the macrophages around alveoli, and these can deal with them and kill the virus. But again, viruses have developed way to escape all these barriers and to reach the bloodstream. Now, as I said, the GI tract is another route of infection, and the barriers are the pH in the stomach. The pH in a horse is about 1 to 2, and yet, some viruses can overcome that acidity. And so, in the gut, the defense mechanisms also are similar to the respiratory tract. So what you have the ciliary movement, you have the peristaltic movement of the intestine, and you have the mucus, obviously the thick mucus. All of these barriers can prevent viruses from enter the gastrointestinal tract. However, viruses also, they develop ways to enter the gastrointestinal tract. And if they do, there are also lymphoid aggregates. We all heard of Peyer's patches. So Peyer's patches are lymphoid aggregates that also can have dendritic cells, macrophages, etc. So they can handle also microorganisms at these locations before they go to the local lymph node, the draining lymph node, which are, in this case, the mesenteric lymph nodes. Okay. Now, some example of the GI tract viruses that can actually resist all the barriers are Coxsackie viruses. They can resist the acidity of the stomach and reach the intestine and affect and reach even the blood circulation. Now, example of viruses that can be carried through the skin are the equine infectious anemia and papilloma viruses. So if we look at the other routes as well, we have the vagina, herpes, conjunctiva adeno, entero. Then in the bloodstream, there is a term viremia, and what we mean by viremia is that the virus is present in the blood circulation. Viremia can be transient in most viruses, so viruses can have just a stage, if you want, in the blood. It can be from few hours to days, and that's a way for blood to spread virus infection, or that's the way for viruses, if you want to use the blood as a way to spread and the infection and reach target organs. It can be also via nerves. For example, in the case of rabies and pseudorabies, the peripheral nerves, rabies virus infect the neurons and can reach the brain,